Hi, this is Mary from Open Helix with this week's Tip of the Week. If you're seeing this tip someplace else, please be sure to come back by our blog at blog.openhelix.com for additional details. I'll provide links to the resources I'm talking about today, links to publications, and to more free training. So check us out at blog.openhelix.com. For today's tip, I'm going to focus on a new feature at the UCSC Genome Browser. If you've seen our materials before, you know we've got training available on the UCSC Genome Browser tools, but they're aimed largely at eukaryotic systems. And there's a new aspect of the UCSC Genome Browser that's become available just this week for researchers who might be interested in the Ebola virus sequences. And because there might be a new community coming in to look at this data, I thought that it was worth taking a quick look at the Ebola Portal and showing you some features of that. So if you go to the UCSC Genome Browser main site, you can still click the Genome Browser link here, and if you choose the group menu and choose viruses, you can access the Ebola genome just like you could any of these other genomes here. And you can find more details about the information that's provided in this browser from this area. But I'm going to show you another way to access this that you might be hearing about, the Ebola Portal specifically. So I wanted you to know that you can get there from the main homepage by selecting viruses from this menu. But there's a separate Ebola portal, which I'll click to now to show you. So here we are at the special UCSC Ebola Genome Portal. And this offers access to the browser. We'll talk about that in more detail in a moment. But there's also other details that are helpful too. So you can find out more on this page, including publications. Some of the publishers have made some of these important publications freely available now. There's more details about the biology, even vaccine development here, and other treatment research is available from this portal. So there's a great deal of information available here. But I thought it's possible that people who are coming to use the UCSC Genome Browser sequences here might not be as familiar with some of the tools. So I wanted to give you a quick overview and point you to more details about that. If you click this first image here with the Genome Browser, you will have access to the sequence information here in a new window, and we will get started using that. So you would click that to load up the browser. And here's what you see when you load up that Ebola virus genome sequence in the UCSC Genome Browser from their portal. So as you can see, there is a virus isolate, this Ebola virus Sierra Leone 2014, and I went over to look at this at NCBI. I took this accession ID here and looked at that sequence. You can also access it from the homepage that I showed before, so you can learn more about this specific isolate here. Now this forms the framework. The A's, T's, G's, and C's from that specific isolate would be the ones that form the framework here in the UCSC Genome Browser, and all the other details are referenced to that. So here there's a lot more detail I'll talk about in a minute. So this isolate is the one that forms the reference sequence. In this area here, you can see the whole length of that virus isolate here, and it consists of under 20,000 base pairs right here. So it's really just a few genes here causing all that havoc. It's astonishing to me. Anyway, so the whole virus sequence is laid out for you right here. You can zoom in though, so all you have to do is click on an item here. So maybe you're interested in this gene. If you click your mouse here and drag across, you can then zoom in. I will cancel this, but I want you to know that that's available for you. What's one of the features here? You can also search for different types of things from the search box, but mostly I'll just focus on the full view that we've got here. But know that you can zoom in and zoom out, just like a regular genome browser that you might be familiar with from other species. The other things that I wanted to highlight here include the features that are available. So in addition to that reference genome sequence that's available here, there are different types of annotation tracks. So these are referred to as annotation tracks that we've got here. And one of the annotation tracks right here is called NCBI genes from a particular GenBank record. And so I will show you how to learn details about what those represent. If you go down to the track control area at the bottom, NCBI genes is the ones that I was just highlighting here. All you have to do is click on that hyperlink to learn more about the source of the information for those genes. It tells you which GenBank record this was. You can link to it from here, as you can see here. Let me go back now and show you some of the other features that are available here. So that was one of the tracks. Another track that's available here is UniProt annotations. This is, also let me scroll down and show you what that is. So the UniProt annotations here are the ones that are shown in dense mode. So that means they're all kind of collapsed together, but let me just click on that and show you what happens if I click on that. So if I click on this track, it will open that track and expand that, and now it's in the pack mode, and you can see that there's actually additional details that weren't obvious when you first arrived. So in addition to the features that we saw before, we now have information about all these other features that are associated with these genes. If you scroll down now to see what we've done, the UniProt annotations now are in pack, but if you click the UniProt annotations link, it will tell you more about the details that are available here. You get information about the types of annotations associated with these genes right here. So let me go back, and now they should still be opened up. And you can get a little bit of information by hovering over them, but you can click on any of these items too. We'll click on this one that's a little bit further out here, and you get more details about the features that are associated with that protein. So those features are available too. Now, more importantly for a lot of people, of course, there's going to be information that they want about the different variants, the different Ebola strains that have been made available. And so, as I mentioned, there's one that forms the reference sequence here, and what you see down below here are other isolates, and the variations from the reference sequence are what's indicated on this browser view. Of course, you can zoom in, as I mentioned before, so you can see here there's a lot of sequences here that have a lot of similarities, but you can see that there are variations here. And if you keep scrolling down, you can see there's even more differences in some of these other ones that have also been brought into this comparison. And the way to learn more about the different ones that are available here is the 160 strains. Let me actually just go back up to the heading that's right here. So what we're looking at is genome alignments, multi-Z genome alignments of 158 Ebola strains and 2 Marburg strains. And so this is 160 sequences, and if you want to learn more about those 160 sequences, you go look at the 160 sequences here that's shown in pack. Again, I'm going to click on that hyperlink to give you more details about what's available here. Here you can see all of the different isolates that comprise that view that you're looking at. And you can turn on or off these different isolates if you want to see those. Maybe you want to turn off the Marburg ones, for example. You can turn those off right here. And there's other things that you can control here. The other feature that I thought was really important is you're going to want to understand the color code, and that's shown here in the display conventions and configurations. So when we were looking at that, you saw that there were some variations that were green, some variations that were red, and those variation color codes are described here. So that's what you're going to want to know about those. Red blocks are polymorphisms in coding regions that change the amino acid sequence. So green blocks are no change to the amino acid sequence. So that's really key. What you want to know is the display conventions and configurations that are associated with all these different sequences. So let me go back. So you can see them, you can look at them all, or you can choose to look at subsets of those. Those are the kinds of features that are going to be important for you. And you'll also see that there's other information here. There's another track that's available for you. This is the immune epitope database. If there are antibodies that have been annotated by the immune epitope database folks here, and those recognize certain regions, those are illustrated on this browser as well. So you can see there's quite a range of information that's available for you here. Not just the genomic sequence that's available, the genes, the features of the proteins that are here, and these variants and this antibody information if that's available as well. So there's a lot of information here, and there's some I'm not showing. When you first arrive at the UCSC Genome Browser, there are white menus that are the ones that are shown on display, but there might be other features that you're interested in looking at too, so you should be able to check those out. And this is the way you can browse around the genomes and look at those, and you can look at the variations here. But I can also highlight the tools menu here will let you access the table browser. So if you wanted big lists of things, you wanted all the variants in certain genes, or if you wanted other features, you can download those from the table browser in bulk. And I'm not going to go into that here. We've got full training on how to use the table browser on our site that I'll point you to as well, but I want you to know that that's available. So not only can you browse around here, but you can query it in more detailed ways with the table browser. That's the overview of the UCSC Genome Browser's new Ebola Portal. Check that out. So I hope that this will help researchers who might be new to the UCSC Genome Browser to understand the layout and overview. I'm going to point you to some more detailed training about the UCSC Genome Browser that's more aimed at mammalian genome sequence, but it will tell you how to use these tools in more detail. This is the quick overview. Please check out the more detailed trainings that we've got as well, and learn how to use the UCSC Genome Browser really effectively to extract the information you want, in this case for the Ebola virus sequences, but there's lots of other genomes as well. Thanks very much for your time.